Hello. The complexity-wise, prokaryotes and eukaryotes differ very significantly. Of course, they carry out in essence, they carry out the same functions or same activities, culminating in reproduction. And that is how they propagate and they last on a time scale. They last long, or they exist in the time scale. Apart from that, in terms of organization, there are great differences between how prokaryotes and eukaryotes differ, and also especially in terms of genetic information. We will try to have a look at how gene expression of these respective genetic information is going to happen. And before that, we will just have a brief discussion on the differences between the gene expression of prokaryotes and eukaryotes. I'm Dr. BCMR. The genetic organization itself is a little bit different in prokaryotes and eukaryotes. Say, for example, the genes are tightly packed in terms of in prokaryotes, whereas they are much more. There is a lot of space between the genes in eukaryotes. Not only within the between the genes, which is intergenic region, but also between the genes or within the genes, which are called as introns. So if we see, the prokaryotes are organized as operons. So this is one gene, and this is another, and this one is another gene. Whereas in eukaryotes, in the operon is a set of genes which are related in function, which have one promoter that is represented as a broken arrow, and that is how usually it is done as. And then we'll also have several genes organized, gene 1, 2 and 3. Usually they are all related to one function. And then there is a terminator. So if transcription occurs, it would have started at the promoter, and this whole region would have been transcribed and then ended there. In eukaryotes, there is a little bit different in the sense that they are usually monocystronic, meaning that one gene has one promoter and one terminator, so the mRNA that is produced will produce only one type of protein. So in this way, it will be a good idea to recollect the central dogma in that sense. So DNA upon transcription will give rise to RNA, and out of those RNA, the mRNA, the messenger RNA, upon translation will give rise to protein. Prokaryotes do not have a nucleus, so everything is together. The nucleoid, which is the genetic material, and then transcription and translation almost happen simultaneously. As the RNA is produced, translation also resumes on the newly being produced mRNA. So transcription and translation are coupled in prokaryotes. In eukaryotes, as we discussed, there are intervening regions which are referred to as the introns. So the coding regions are referred to as exons, exon 1, 2, exon 3 and exon 4, and there are 3 intervening sequences or introns. So for a protein to be produced, these introns should be removed and the exons should be joined. So in eukaryotes, we have a nucleus where replication and anything to do with the genetic material happens within the nucleus. That is, replication, recombination, anything happens that will be in the nucleus, because that is where the DNA is. And transcription also happens there because DNA is the template. And then the produced pre-RNA could be the removal of introns and joining of the exons happen, and that is when the mature RNA is formed, and that is mature mRNA that is transported out into the cytoplasm where translation occurs. So the point that we need to remember is that the splicing is an important aspect in eukaryotic transcription. So transcription as such is a template dependent process, like many other processes that happen within the information within the cell, because the genetic information is present as a sequence of nucleotides, and that expression of those nucleotides should happen and produce corresponding elements such as proteins or enzymes. So everything, the ability to read the template and produce or translate that information into a phenotype is template dependent. So transcription is carried out by an enzyme called as RNA polymerase. That is, RNA polymerase is a DNA dependent in template wise and DNA dependent RNA polymerase. It polymerizes the substrates R and TPs. So the important aspects or differences between prokaryotic and eukaryotic, transcription wise at least, is that in prokaryotes the mRNA is polycistronic, which means that there are more than one open reading frames within the mRNA. They can be, if not all of them, majority of them. The organization is in terms of operons, so one mRNA has 3 open reading frames and each of them will produce a protein. In eukaryotes it's usually monocistronic. I hope we will go through, with these fundamentals, we'll go through the lessons of transcription or the discussion, and we'll try to understand them at a deeper level. Thank you very much.